So there is the lab folder here. And then if you click on answers, the lab answer for the first lab, right? For the first PowerPoint is now available. So compare your own answers with that. If you have not done the lab yet, I strongly encourage you do it yourself first before looking at the answers. Okay, it's a good idea to kind of test whether or not you know this stuff. So for the lab quiz, right? Yes, there are going to be these models, but in addition, there will also be standard multiple choice question. Think of the lab quiz as like kind of like a review for yourself, right? Just like the do it when you finish studying. I wouldn't do it as the first thing. But do it when you feel that you've done enough studying and you're ready to kind of test yourself. And then you have 2 tries anyways, right? So
Like this picture, Professor, I was confused between the superior lobe and the right lung, which will be the right answer. So, but you have put both of them. That was
right,
one of
yeah,
my
yeah. So
questions
like
last time.
on the test, right?
Yeah.
On the quiz rather, the lab quiz, I'm not gonna have 2 choices where one choice is right lung and then the other is superior lobe, right? Like I'm not gonna trick you like that, right? So
Okay.
it will either be one or the other. Okay.
And then the other thing I was thinking, because this is coming from after the respiratory, most of the things are respiratory, and I was thinking for why is it aorta or is it maybe pulmonary?
Right.
Um,
Yeah, pulmonary artery, right? But
yeah,
pulmonary artery is this one, right? Where my mouse is. You kind of have
exactly.
to see where it's connected to, right?
When I
Um,
when I saw in the color, then I did realize it, but because
right.
it is red color, so it cannot be pulmonary artery.
That's right. That's right. Okay. And so that's why, having the answers here is gonna help you guys as well. It would be much harder, right, if I don't provide the answers. And that's why I'm doing it for you.
I
Okay.
will say impossible, Professor.
Was that impossible? No,
Yeah.
I wouldn't say it's impossible, but it would take a little bit more effort. Okay, so if you don't already have the review, please get yourself a copy. Um, and if you don't know where to get it, just go to the homepage here, and there's a test review, right? Okay, test one review. So like I said, no class on Wednesday. Don't come. Okay? 75 minutes should be enough, okay? It should be enough. It's not a lot of questions. There's 30 multiple choice, and then so like 40 questions, right? 40 questions. I think it should be enough. Right? Yeah, and it's only lecture 2 to 4. The lab quiz, however, is gonna include the lecture 1 as well. Okay, do you guys understand what I'm saying? I know it's very confusing. You
Yes.
have a lab quiz tomorrow that's due tomorrow, right? End of tomorrow. Okay. That one's 2.5%. Lecture 1, 2, 3, 4. Okay. 2 attempts, best one counts. On Wednesday, no class. You can do the test. The test is 20% of your final grade. All right. Okay, fantastic. Let's get on with the review sheet. There's a question. Is the application same as the long answer question? Uh maybe, I mean, like, it could be, but it doesn't have to be, right? So like we oh, did we do oh, thank you for bringing that up actually. Okay. We have the lecture 4 application question that I have not taken up yet, right? So let's go through those first. Um and then we'll do the review. But yes, like some of the review questions, some of the application questions will be similar or will be kind of like these application questions, but some you will have not seen before. Um, yeah, but again, it's not impossible. All right. What are you guys? Okay, like I'm open. Like, does 75 minutes seems reasonable to you guys for like 40 questions? You can be like, you know what? Why don't I know some of you are always thinking, oh, more time is always better, but not true. Hey, sometimes like if you have more time, then you end up, like thinking too much about certain things, and then you overcomplicate things. Like, what do you guys think? Let's do a poll. Let's do a poll. Okay. Like, do you think 75% is adequate? A if you think is good, and then if you think more is better, please choose B. Yeah, I'll see what the class think. Sometimes you just, you dwell on it, right? On the test, and you're like, ah. Okay, so for those of you who are thinking 75 is not enough, what's a good number? Give me I'm open to ideas. What is a
90.
reasonable? Give me a number. What did you say?
90.
90? Anyone else? 80 to 90?
Professor, I'm full stress every test.
Yeah, I'm stressing you out
I
every
need
time.
the first 20 minutes. I have to overcome in my stress. After 20 minutes, oh, I'm relaxed. I can do
Right.
my best.
Yeah, okay, sure. I'll give you guys 90, whatever, it doesn't matter. Where is this? This assessment quizzes. What are we talking about here? Test 1, right? That did time limit. Here, 90, okay? Now everybody's happy. Save and close.
Thank you, sir.
Yeah. Okay, done. Any other requests? Other than, telling you what's on the test? I mean, you kind of know already. Like, I think I don't think you should be surprised by the questions. Like, I mean, even though quiz 1, right? Some of you were a little bit, I don't know, like taken back, or like, thought it was a little bit harder than you thought. But if you really think about it, most of the questions are, like the things we did in class. Sure, there are a few that requires thinking, but that's how it is, right? Like, it's an online environment, right? Not everything should be, textbook answers. Otherwise, is wouldn't be an accurate assessment of what you know, right? Okay, so hopefully you will find it to be reasonable this time as well. In any case, we should get started. Melisandre learns from the internet that by deliberately hyperventilating, inhale and exhale rapidly, she was able to extend the time she's able to hold her breath. She wants to impress her friend, but with this, so she hyperventilates for several minutes before diving into a pool, thinking she will be able to swim farther by holding her breath longer. Shortly after she enters the water and begins swimming, she blacks out and almost drowned. Explain why she was able to hold her breath for longer on land.
I'm sorry, sir.
What's that?
Sir, do we have any diagram in our first test?
Do you have any diagram in the first test?
Yes, sir.
No, most of the diagrams is going to be in the lab quiz. Okay. I mean, there might be, but it's not the focus. I cannot promise you won't have any, because like I said, there's a lot of questions in the bank. I don't
want.
You don't want diagram?
Yes.
Yeah, I can help you with that though. Like, it's in the bank somewhere. Okay, like you might get it, you might not get it. Okay, but the lab quiz is all about diagram anyways, right? Okay, like if you don't know your diagrams, if you don't know your labels, you won't be able to do the lab quiz, right? So you have to study for them anyways. And I would assume it's a transferable skill on the test, right? Okay. So here, we talk about this, right? By hyperventilating, you basically, there are a couple of key things to know, to remember here, okay? Like CO2 is the gas that stimulates the brainstem, right?
I think the medulla oblongata.
That's right, the medulla oblongata. Okay, that's where the respiratory center is, and it's sensitive to carbon dioxide, and that's gonna, give you the urge to breathe. Okay, so when you hyperventilate, you are getting rid of carbon dioxide that you normally would not be getting rid of through regular breathing. And by having a lower carbon dioxide level to start with, when you hold your breath, it takes longer to reach the threshold, right? Remember this graph we drew last class? There's the threshold, and if you're just breathing in and out regularly, this is CO2 in blood, and this is time, right? It's gonna take some time to get to the threshold, and then you have to breathe. But by breathing out all the excess carbon dioxide, your starting level is lower, so it takes longer to get to the threshold. That's why you can hold your breath for longer, right? But you're not getting extra oxygen in your system, and so when you are diving and swimming, after doing this, you are basically using oxygen that you don't have, right? And in that case, you don't have enough oxygen going to your brain, and you black out. And underwater, when you black out, you breathe, right? You breathe in the water, and that could be a dangerous thing. Okay. We also talk about this scenario. You can read it later. This talks about the kid who's holding the breath and eventually lost consciousness. So is it a dangerous thing? Look, I mean, it's not ideal, but as soon as the person loses consciousness, they will start breathing. Their respiratory center will override the voluntary inhibition. Okay, so as soon as the breathing comes back on, they should be okay. Okay, so that's that. I mean, these are not super hard questions. We'll do a few harder ones in the review today, actually. Hopefully, it will be it'll help you. Okay, so there is a test one review. Let's get to it. Again, feel free to stop me if any questions come to your mind, or like if you want me to elaborate on anything specific. Okay, so now this is just like a general review, huh? Okay, like I try to put everything that's important here, but it's by no means the only thing that you could be tested on. It's a fairly good starting point. I would say if you know everything that's on this review sheet, you should get a good grade. But if you want to get perfect or in the 90s, you might have to go through the notes again on your own. All right, so this is blood that has been spun down. What is the top portion here? What do we call that? Anybody?
Plasma.
The plasma. That's right. So let's write that down. This is the plasma, which makes up approximately 55% of your whole blood. We know it is mostly water. With dissolved ions, nutrients, and wastes, and proteins. Lots and lots of proteins, right? So let's talk about some of these plasma proteins. We have learned a lot of plasma proteins since since lecture 2. All right, so at the time we learned that there are things like albumin. You might remember albumin is important for maintains osmotic pressure. In other words, it keeps the fluid in the blood, right? Otherwise, it's gonna start leaking out of the capillary, and that could create swelling, right? An accumulation of fluid. And we talk about clotting protein. Does anybody remember the clotting protein names?
Fibrinogen and
Thrombin?
Yep, fibrinogen and prothrombin. Thrombin is the activated form, so the prothrombin. Okay. So make sure you know the pathway, okay? Know the coagulation pathway. You know that one that has the thromboplastin. I'm just gonna show you a picture of it. I know there is a question on that.
goes thromboplastin and then goes to
Yeah, like
prothrombin
this
and
one.
then thrombin and then yeah.
Yeah, that one. Okay, so make sure you know the steps. I'm pretty sure there is a question that's on everybody's quiz test, where we give you the steps and you have to rearrange them. All right, so make sure you I know it's not filled in here, but you know it's in the class notes and it's in the study guide answers, right? So please have that with you. Okay, so that's a plasma protein. And in terms of immunity, like plasma proteins related to immunity, at the time when we learned this in lecture 2, we wrote antibodies, right? So we now know the antibodies are created by like B cells. Does anybody know any other proteins that we've learned later on, like in lecture 3, for example, that are related to immunity and it's in your blood?
Competent protein.
Mm-hmm. The complement proteins, that's right. Complement proteins. Okay. Now I'm just gonna verbally remind you what it is. I don't expect you to write down everything that I say. It's like I know I'm going too fast for you to write down. But if you don't remember what I'm telling you, then you should put a star next to it, and then after class you go look it up yourself, right? Okay, so complement proteins, these are made in the liver. They will help. The word we used in class was synergize, which means it works with other immune response to make it better. So they leave chemical trails for cells to follow, so they know where to go. They will tag the pathogens, so it's easier to perform phagocytosis on them. And they also can form these things called the membrane attack complexes, right? These membrane attack complexes will punch holes in the bacteria. Okay. I don't know if you know this, but you can also turn on captions, right? Like the things I'm saying to you there are captions that are generated live, right? So if you have trouble following, you can turn them on, and it would make it a little bit easier.
How to do that, Professor?
Do you have a ribbon? I cannot show you because like the screen won't capture this the
Okay.
Zoom
Yeah, I
stuff.
got it.
You got the yeah, you turn it on now?
Yeah.
Okay, yeah. So go to the ribbon, press more, and then captions. Okay, some of you will be able to do that. Okay. All right. So that's the plasma protein, huh? Which of course is in the plasma. Now, this middle layer, the middle layer is called the buffy coat. The buffy coat is very, very thin, and it consists of platelets and white blood
Leukocytes?
cells. Yeah, leukocytes, white blood cells. This is only about 1% of all your all your blood. Okay. And platelets, of course, it plays a role in blood clot formation. They come from a bigger cell called the megakaryocyte. Yeah, thank you, Madison, for posting that information. Yeah. They are fragments from this cell called megakaryocyte. Okay. No nucleus, right? Okay, the red cells and the platelets, they don't have nucleus.
Professor, I have a question.
Mhm.
Is the erythrocyte includes the Buffy coat or not include the Buffy coat?
No, the red cells it's all at the bottom. Yeah, not in the Buffy coat. No, all this red stuff down here, that's the erythrocytes.
I mean the formed elements, the form elements.
Oh, form elements, yes. Form elements
That's
include,
what-
yeah.
Yeah.
Okay, so the Y cells, people. Y cells, we have two categories, remember? There is the granulocytes. And again, if you have trouble memorizing which ones are which, it's okay to have the chart in front of you, right? Like when you do this. Okay, it's okay to have a little bit of help from your notes. Okay, so granulocytes, we talk about neutrophil. Neutrophil, we talk about eosinophil. All right, we talk about mast cells and basophil. I know at the time we did not put mast cells, but we learned it later on, right? We learned that the mast cells and the basophil, they are related, aren't they? Right? They both play a role in inflammation, right?
But
As
Professor, are they part of the white blood cell or they are you said they are anchored in the tissue, the mast cells?
yes, but remember, white cells is special compared to like red cells because they can squeeze themselves out of the capillary and reside in the tissue, right?
Okay, got it.
Yeah, yeah. So agranulocytes, we have things like monocytes, which can transform into macrophage, right? If you don't remember what these things do, that's okay. I have more questions coming up that summarizes them for you. Okay. There are the lymphocytes, which is part of the lymphatic system, and we now know there are T cells as well as B cells, right? And even under T cells and B cells, there are different kinds. Right? So if when you are studying, okay, create your own flowchart to organize these cells. You can organize the cells based on, in this case, based on appearance. Okay, granulocytes have the granules when you look at it under the microscope, and then the agranulocytes, they don't. Or you can group them based on function. Which one of them are phagocytes? Which one of them plays a role in inflammation? Okay, so you can group them using different categories.
Professor, why don't we say them here naive T cell and naive B cell over here?
Yeah, I mean, there's no particular reason.
It's
Like,
the same thing, right?
yeah,
Okay.
yes and no, but because like there are so many T cells, right? They have the naive T cells, but then they can become helper T cell, killer T cell, suppressor T cell, all these different
Right.
T cells, right? So like
Okay.
we just kind of drop the adjective and just call them T cells in general, right?
Okay.
Okay, and then we have this like big portion at the bottom. That is the red blood cell. Red blood cell. And this particular code, sorry, I should go with the same naming system here. So this red blood cell portion, that's the hematocrit. Hematocrit, which is, contains RBC. RBC. Right, so I'm gonna put a star here. Right, these things are called formed elements, the cellular component of blood, basically. Formed elements. And I should also remind you, they are produced in bone marrow, okay, through, I don't know if you remember,
Mitosis?
Not quite, but good try. Hematopoiesis.
Hematopoiesis, yeah. Professor, sorry.
Yeah, no worries.
You will be posting the video afterwards, right?
Yes. I'll post this
Oh,
video.
okay, because I always just read your. I don't write when you are giving the lecture, and I do it after I review it.
Yeah, sure.
I
Whatever.
make my notes when
works.
I review it.
That's right. Yeah, different people have different strategies. I'll post it. I'll post the notes as well. Okay, as I always do. Okay, so that is the hematocrit, the RBC. Couple of things that comes to mind: no nucleus. Contains hemoglobin. If I ask you, where do you normally find hemoglobin? Inside the red blood cell. Okay, you're not supposed to have hemoglobin in your plasma. Okay, they only come out if your red cells are dying. That's not a good sign. Okay. They are biconcave shape. Okay, why? Because you need to increase surface area for gas exchange. BBS, do WBC have nucleus? Yes, they do. Okay, only the red and the white, only the red and the platelets don't have nucleus. The white ones, they do. Sometimes they have more than one, actually. Lifespan will say approximately 120 days. Okay, and you can break them down. Breaks down at liver and spleen. We also talk about how your kidneys make EPO, right? Remember that one? EPO is
It increase the efficiency for making the white blood cells.
That's right. Increase red blood cells. Red blood cells. Okay. That is basically what I can think of so far.
So when you say lifespan 120 days, you mean that's how long they can survive?
Yeah, approximately. Yeah, approximately 120 days. Yeah. So after, like, your bone marrow continuously make new ones, right? Okay. So like the ones that you have now, okay, that were made, in December, they are probably like, at the end of their lifespan. So they will get recycled, broken down in the liver and the spleen, right?
Thank you.
Yeah.
Sir, but outside of the body, they can stay up to, like you said, 6 months, right? Like,
I don't think so. If you store them in like a fridge or something, I think they're good for not 6 months, like maybe I think several weeks to a month. The plasma, however, you can store for a very long time, like 6 months to a year, I think. Yeah, plasma has no cells, right? Plasma is just the liquid portion, right? Okay, now here is a possible application question. The trick is this: if I tell you the answers ahead of time, like in class, then it's not really an application question anymore, right? Because now you are not thinking about it. Now you're just, remembering what we talk about in class and then, writing down the answers. But that's okay. Like this would be an example of an application questions that you might see. Okay, like in another context, like you would have not seen the question before, but it's the same level of difficulty, same level of complicatedness that you have to think through yourself. So like I show you a picture of this, and I actually compare to two test tubes, explain what might cause the results on the right. Okay. Anybody?
Maybe not properly centrifuged, or maybe a little bit got moved after centrifuge.
Okay, so Mohammed said it's not properly centrifuge. Could be, but anyone else? Like if I tell you that this is some kind of it's not a technical problem. It's not because of the technique. but some kind of conditions. What do you think this is? Tohib, you want to say something? I see you on mute.
Like, it might be caused by addition of chemicals that react with the original clear solution.
Okay, okay. So like you guys are still thinking it from a perspective of, you know, something that we do to the blood. Okay, but that's not what I'm trying to get at here. Like what kind of conditions, what kind of abnormalities in the body?
It can
Maybe the
reaction
less
to insoluble precipitate.
Insoluble, what, sorry?
Precipitate.
Okay, for some kind of precipitate, all right.
It's albumin is less in plasma.
Sorry, a woman is what?
Albumin.
Oh, albumin. Okay, low albumin in the plasma. Okay. Sarah thing is anemia. Okay, okay. All these are good suggestions. Anyone else? Blood thinner.
Or
Okay.
like coagulation, like a blood clot and or something like that, or like for example the RH incompatibility.
Okay, now we're getting somewhere. Now we're getting somewhere. Okay. So I hear RH incompatibility. Although that's not related to blood clot though, right? Blood clot is like your blood, you know, clotting, forming like chunks, right? So continuing that line of thought suggested by BB, RH incompatibility. What is the word I'm looking for, people? Come on, help me out. I'm just looking for one single word that would explain this situation.
Sensitized.
Okay, and then what? And then what? And then what's going to happen to you, to the blood?
Start bursting.
Start what? Bursting, did you say?
Yeah.
That's right. Okay, so this here, this is normal. Okay, normal. Okay, this here, this is hemolysis. Okay, that's why there's red stuff in the plasma. You're not supposed to have red stuff in the plasma. What makes your blood red, guys? Do you remember? What is the thing in your blood that makes the blood red?
Red blood cell.
Okay, but what is in the red blood cell that makes your blood red? We talk about this.
Hemoglobin.
Hemoglobin. And what did I say earlier? Where are you supposed to find a hemoglobin?
In the red blood cell.
In the red blood cell. And right now we have the hemoglobin in the plasma, right? That tells you your red cells are bursting right and left, right? And then you got all these hemoglobin coming out and they're in the plasma. That's why the plasma is red now. Understand? So that is what hemolysis look like. This is for real. Like if you collect blood from a patient and you spin it down and their plasma looks like this, we got a situation going on in the body. A lot of blood cells are bursting. Why? What are some reasons? Well, people already suggest it could be like, you know, if this was taken from a kid, then maybe there is some kind of RH mismatch going on, right? But if it's taken from an adult, it could be like a mismatch blood transfusion. Right? There could be some kind of internal bleeding. Okay. There could be maybe some kind of adverse reaction to some kind of poison, some kind of toxin. The key thing I'm trying to get at here is hemolysis. Yeah, this is very dangerous. Like you gotta find the cause right away and then try to fix it. Right? If it's like a mismatch of blood transfusion, you gotta flush it out with the right kind of blood. Okay. Does that make sense? Right? So like this requires a lot of thinking, right? It's not just regurgitation of what we learned in class. You have to understand the stuff and apply it to a situation we have not seen before. That's why it's an application question. But don't worry too much, right? Like there are only how many? 5, I think that's
5.
what I said.
5,
right?
yeah.
Yeah, 5, right? So like it's not the end of the world that you don't get. It's only 1 mark each. Okay. So just try your best and you will be okay.
I have another question, Professor.
Yeah.
The other tube should be having 55% plasma and 45% formed elements.
I know, but sometimes it depends on what's in the tube that you're collecting it. You actually can collect it in different color tubes and they do different things to it. And that's why the portion doesn't, look quite right in this case. All right. So I hopefully you understand why there's a need to put some of these harder questions on, right? If not,
So I have a question.
yeah.
So when they collect the blood, at what point does it separate?
So they have to put it in a centrifuge, right? The machine that spin it at really, really high speed, right?
Okay.
But like I said, there are different tubes that they could collect it in. And the different tubes have different functions. Sometimes sometimes you actually want the blood to clot out, right? Then you would collect. You guys have blood taken from you before, right? Like you notice the cap is different, right? Sometimes it's lavender, sometimes it's like the minty color green, sometimes blue cap. They actually all have different meanings, and it depends on the application of it. The most common one is the purple one, the lavender, which is just like collecting blood, and later you spin it down to separate it and analyze it. Okay, you can look it up. You can look it. They are called vacutainers, and you can search up the cap color and what they mean. All right. Next, match the parents with their babies. There will be babies will be lost on the test, and you will have to match them. Okay, that's part of the questions. So make sure you practice with these questions. So to do these things, you need to remember the 4 things, right? Type AB blood cannot have type O child. We wrote this down already. Type O cannot have AB. And then negative will always have negative babies. And then if both parents are type O, they will always have type O babies. So in a question like this one, we always start with a parents that have AB or O, and even better if it's AB and O, right? So right away, I can eliminate a lot. Right? It's not this one, it's not that one. Because again, AB cannot have O, and O cannot have AB, right? That's why I can cross out the first 2. They are both negative here, which means they cannot make positive. So this is their baby. Baby K is their baby. Right? Does anybody have any question about that first explanation here?
No, that's good.
Yeah, the rest of the class? Okay. All right. So then we pick something else. We're gonna pick like this one maybe. And there's a type O there, so not the first baby. They are both negative, so not B positive. So it must be baby I. So baby I is gone. And do I do the second one or the third one? Well, always do the ones with the O or with the AB first. There is an O here, so AB is not their baby. It has to be J. And then H is the last one. All right. So obviously yeah.
You said negative and negative can't make a positive baby, right?
Cannot. Yeah, cannot.
Cannot.
Yeah.
Right. Okay, so positive and positive, that will also mean it can only make a positive.
Not necessarily, because a person can be positive, homozygous positive, and that would be Rh positive, right? But the person can also be positive / negative heterozygous, and it will also be positive, right? So if I have two Rh positive individuals who are both heterozygous, and if you do the math, if you do the Punnett square, right, there is a 25% chance that they're gonna have a negative baby, right? It's only when they're negative that they are homozygous and that they're guaranteed to have a type O baby, right? Is that all right?
Yes. So for example, can like let's say an A plus and a B plus, there's like what baby will they have?
Now, so like based on the A and the B, right? We cannot say for certain because it depends on whether the person is like IA or IA small I, right? But the point here with these type of question is there are a limited number of choice of babies that belong to them. That's why we are able to, you know, eliminate certain numbers, certain babies, right? So we're not saying this couple can only make like AB negative, right? Like certainly there are other options as well. For example, if the A and the B, they are like IA little I and IB little I, then they could also make type O baby as well. Okay, so like we don't know, like could this couple make O negative? Absolutely, right? If they are IA small I, IB small I, and they are both heterozygous, they could also make O negative. Okay, but then the question here, that's not what the question is asking, right? It's not asking what kind of baby they can make, right? It's asking, you know, if we have these kind of babies, who are the parents? And O negative is already taken by another parent here, right? And that's why we know it has to be the AB negative. Okay.
So, Professor, we should start with the people who has O group, right?
I would start with anyone with O or anyone with AB, and if there is a couple that is AB and O, that's definitely the one I start with first, because then you know you want to start with the parents that allows you to eliminate the most like most of the babies, right? If you can eliminate a lot of it, it narrows down to one. That's how you can approach these questions, right? All right, here is another example of an application type of question. Yeah. And again, we're doing it now, so you can prep yourself a little bit. Now, don't think like this, right? Sometimes students have these weird psychology, and then I don't know what's going through their mind. They're like, "Oh, he did it in class already, so it won't be on the test." Not necessarily, okay? I'm doing it in class because it's probably going to be on the quiz, okay, on the test, right? So you know, make sure you spend some time to understand it. Okay, so here, a new blood type called fluffy has been discovered in humans. I just make this up, right? There are three, sorry, there is a typo here. There are three alleles in the fluffy blood group, F1, F2, F3. If F1 is dominant over F2 and F3, and F2 is dominant over F3. Okay, so basically F1 is dominant over F2, which is dominant over F3. That's what it says, right? Which means 1 will cover up 2 and 3, and 2 will cover up 3. How many different phenotypes are there in the fluffy blood group? Well,
Three.
we will see. Let's write down the genotype first. Okay, the genotype.
Is it 3 factorial?
Is it 3 for the answer?
Factorial.
3 factors, 3 alleles. Yeah, 3 alleles. Okay. But let's write out the genotype first. Okay. The genotype is the genetic combination. And so you know there's F1, F2, and F3, right? So a person could be, you know, F1, F1. Right? You know, I'm just gonna keep it simple. A person could be like 1, 1. Could be 2, 2. 3, 3. Right? In other words, like F1, F1, F2, F2, F3, F3. What other combinations are there? Please help me out.
1, 2, 1, 3.
Exactly, right? It could be 1, 2, 1, 3, and one more, please.
2, 3.
2, 3. And that's all, right? There are 6 possible genotypes. 6 different genetic combinations, right?
Yeah, 1, 3, 2, 3, and 3, 3 will not be 1.
What's that again?
1, 3, 2, 3, and 3, 3.
We have 1, 3 already though, right? 1, 3.
Oh, okay.
Okay. So what about a phenotype? Well, let's take a look. Okay. The first one here, 1, 1. What kind of blood group is that? Well, it's only 1. So then we'll call that, fluffy 1. Fluffy 1. The name doesn't really matter. Like I'm just like trying to, it's easier to see it's 1, right? So that's fluffy 1. Is there another combination here that will be fluffy 1?
No.
You sure?
Fluffy
Oh,
2.
yeah. 1, 2, 1, 3.
Exactly. 1, 2, and 1, 3. Why? Because the question says 1 is dominant over 2 and dominant over 3, which means whenever you have a 1, it will cover up the other ones, right? That's fluffy 1. Okay, how about this one? What is this guy? 2,
Fluffy
2.
2.
Yeah,
2,
fluffy
2,
2.
2, 3.
That's right. That's correct. Both 2, 2 and 2, 3 will be fluffy 2 because 2 is dominant over 3, according to the question. And finally, if you have 3, 3, that's just fluffy 3. Okay, so there are the answer is 3. There are 3 possible phenotypes for this newly discovered blood group. Okay, now I'm gonna ask you to try the next one, and I'm gonna give you some answers, and you're gonna do a poll for me. All right, take your time.
Excuse me, what does mean condiment?
Codominant means
Yeah,
like IA and IB are codominant. They both show up.
okay. I got it.
Okay, anyone else? I guess I'll give you a few more seconds. Okay. Well, let's go through it together. So in this case, all 3 alleles are codominant with each other. Right. So I have 1, 1, 2, 2, 3, 3 like before. The genotype doesn't change. Right? Genotype doesn't change. It's the phenotype that's gonna change, right? So look, all of them are going to be different. Like that's gonna be like fluffy 1, fluffy 2, fluffy 3. And then this one is like fluffy 1, 2, fluffy 1, 3, fluffy 2, 3, because they're codominant. Codominant means they both show up, just like our AB blood group, right? If A and B are there, they both show up, then we call it type AB. Not just A, not just B, but AB. So if 1 and 2 are together, then we call it like fluffy 1, 2. Right? So for that reason, the answer is 6. Anybody have any question before we move on?
Is there a way to calculate this before instead of like writing it down?
No, this is purely logic. Yeah, there's no formula or anything like that. Like you just have to, each scenario is different, right? So like I can, for example, I can say F1 is dominant over F2 and 3, but 2 and 3 are co-dominant, right? And then that would be different, right? Okay. So every scenario requires you to think this through. Okay. All right, let's do the Punnett square.
Professor, I made a mistake in Punnett square. I don't know why I did that, but when I made the Punnett square, I made F1, F2, F3, and then F1, F2, F3, and the both sides made me 9 Punnett squares. Nine genotypes. What mistake did I did?
For the genotype?
Yes.
Oh, I because 1-3 is same as 3-1, right? That's probably why.
Yeah, but when I did when I did it, I
Uh-huh.
made 1, 2, 3 on one side, 1, 2, 3 on the other side. That gave me 9 options. Yes, the possible outcomes are from there,
Yeah,
but
but that's what I'm saying, right? In your table, you probably have 1-3 and 3-1 separately, right?
yes,
But they are the same thing, right? So you double count some of it, right? Because the
Correct.
order doesn't matter. The order
Yes.
doesn't matter, right? That's why. Does that answer your question?
So what, like, I don't know what is my where I went wrong when I made 9 instead of 6.
You didn't do it wrong, but you count the same thing twice. That's where the mistake is, right? You should have count 1-3 and 3-1 as the same thing.
Oh, okay,
Yeah,
got
just
it.
like AB, right? Like IAIB, same as IBIA, right? They're both AB. The
Yes.
order doesn't matter. Yeah.
That's right.
Okay. All right. Here is another question. We're supposed to find the genotype of question mark. Right, these people. All right. So, obviously, when you do something like this, you gotta do it on paper. Okay. Don't try to figure it out in your head. Don't try to do it on the screen. You gotta do it with a physical paper. That's what I would suggest. So you write down the genotype of A, B, and O first. Those are the ones that have only a single genotype, right? There's no other way for someone to be type O other than little I. And for type A, it is going to be IA something. We don't know what the other one is.
Small I.
Yeah, maybe, but like, when we're
Sure.
doing this question, we don't know yet, right?
Okay.
So like, I'm just going through the steps of how you would approach this. Okay. So that's how you would set up your answer to start the question. Okay. No thinking is required here yet. You just write down the genotypes. Then you got to start using some logic. Yeah, you got to start using some logic. So for example, sometimes you have to chase down the generation. Sometimes you have to chase up the generations. It depends. There's no, fixed way of doing this. That's what makes it a little bit challenging, right? So like there is a little I here. So it has to come from one of the parents. Okay, so I'm going to put the little I here. And then there is the other little I, which has to come from the other parent, right? Okay. That's for certain. Next, where does this A come from? Is it from the dad or is it from the mom?
Any
Mom.
one of them, that would be from the mom.
It has to be from the mom. It has to be from the mom, right? Because look, if you trace back one generation further, these grandparents don't even have an A. How do I know they don't have an A? How come I know the other one is not A? Well, because if it's A, then their blood type would have been like AB, right?
Yeah.
Which is not. So the IA has to be from this pair, has to be from mom. Okay, and that means the IB would be right here. Okay? So this one is not too difficult. So remember the Shrek one that we did in class? Okay, so review that. Review this one. I think there is another one here that we did. Did we do this one? I don't remember. Can someone tell me if we did it together?
I don't think so, but it's easy to do.
Yeah. So like there's this one that you have to do. If you don't know the answer, look at the review sheet. I'm sorry, the study guide answer. That's as hard as it gets. Okay, if you understand all the ones we've done in class, as well as the ones in the study guide and the review sheet, you should be fine. There are some
These are
poll
my
questions
favorite
as well,
questions.
right? So was that these are your favorite question?
Yeah, most easiest ones.
Yep, there are only one or two though on the test, okay? But what I'm trying to say is, if you're able to do these ones, you should be okay. I don't want you to get overwhelmed by and try to find more questions online. You don't need to do that. Okay. All right. So these are some of the longer multiple choice questions, right? They are not your typical, factual stuff. So please give it a try. Okay. Let's take it up. I only got half the people answering, but that's okay. We got to move on. All right, so let's take a look at the choices and see if there's anything wrong with them. A drop of Sarah's blood was mixed with anti-A antibodies. Another drop mixed with anti-B, and then the third mixed with RH. So that's like the lab stuff, right? That we talked about. I don't know if you remember seeing this. So this is anti-A, anti-B, and then anti-RH. And clumping was not seen in any of the tests. So no reaction, no reaction, no reaction, right?
I guess it must be negative O.
O negative, that's right. So this person is O negative. That's excellent. But let's take a look at what's going on here with the choices. The test must have been defective because clumping should have been seen in at least one test. That's not true, right? It's possible to have no clumping at all. All right. So O negative. Now in real life, you would have to have like a control in your experiment, right? In your experiment to make sure that you're not actually doing this wrong, that clumping is actually possible. But it's okay. It's okay to have no clumping. For someone who's O negative, obviously no clumping. None of the antibodies in Sarah's plasma reacted with the antibodies. That's not true either. We are not looking at whether or not the antibodies are reacting with each other. We're trying to see if Sarah's RBC is reacting with the antibodies, right? Okay, it's the antigens that are on the surface of the RBC that's doing the reaction, not the antibodies. Sarah's blood type is O positive. Nope. Right? If it's positive, it would have been reactive with the RH, but it didn't. It's O negative. Sarah does not have any ABO or RH antigens on her RBC. That's correct. That's the right one. Okay, so nobody chose D. But I hope I have explained to you why it is D. Okay. Any follow-up questions?
I don't understand how it's O negative instead of O positive.
Okay, there's no clumping, right? Not even with the anti-RH. If the person is positive, then the antibody would have reacted with the red cells, right? But it didn't. The fact that it didn't react with the red cells means there's no positive. It's not positive for RH. No, is that like, for example, if the cell has RH, the anti-RH antibody is gonna attack it, right? But we didn't see the attack. We didn't see the clumping. That means there's no RH, right?
Oh yes, because the RH is, you can only have RH if it's negative.
No, you're thinking
Right?
about this backwards. You're thinking what kind of antibody you're gonna make, but that's not what this is about. The antibodies are already in the plate. These antibodies are not from Sarah. These are just antibodies that are already in the plate. And we are trying to see if Sarah's blood is gonna react with them. Okay. So when you add Sarah's blood to the well, and there's no reaction, it tells you Sarah does not have A on her red cell. Okay, we are not trying to think about whether Sarah is making the anti-A antibody. That is not what we're talking about at all. Okay, so review the recordings on the blood typing stuff, and there are more questions here that you can do in the review sheet. Review, what do we call these things? The study guides, right? Question 13 and 14. Okay. You know, just for safety, let's just go through a few of them. Okay. Look at the first one here. Okay. Yes reaction with A, no reaction with B, yes reaction with RH. You know, BB, why don't you try this one if you don't mind? I mean, you don't have to, but like, you know, if you want to give it a go.
Sure. 1 second. So anti-A means it can't, like, it would be B.
It will be B. No, we are, again, you're thinking what kind of blood type makes anti-A. That's not what we're talking about. We're talking about, we already have the anti-A in the well, and you're trying to see whether or not the blood is reacting to it. Okay, so the fact that the sample reacts with anti-A tells you the person has the A antigen. No reaction on B, so no B. Reaction with RH, so positive. Okay, you will see a lot of these questions in the lab quiz. Okay, the lab quiz. Okay. All right, next one. Try another one. Anyone. Okay. Anyone.
B positive.
This is B positive. Reaction with B. Reaction with RH. That is B positive. All right, so try it out. Check the answers in the study guide if you're confused. And you know, also watch the recording. Okay, let's go back to the test one review here. Okay, let's do one more, and then you know what? I think we should take a break. I was overly ambitious to try to get it done in an hour and a half. We're not going to be able to do that. So select all, save blood transfusion.
Should we consider sensitizing or not?
We always try to avoid sensitization. Okay, so positive to negative is never okay. Okay. Even though like theoretically it's okay once, right? We talk about in class, but like, you know, we assume we don't want to sensitize. Okay, let's leave it at that. So you can use the transfusion cheat sheet, right? Call it help sheet if it sounds better. But don't worry about having this next to your computer. Yeah, it's useful. So O positive, A positive, not okay. Positive cannot give to negative, right? AB negative to AB positive, that's okay. That's safe. Same blood type, negative to positive, no problem. This one's also okay. You can see A to AB is okay. Positive is okay. B negative to AB negative is also okay. And then this is not okay. So B, C, D are okay. All right, let's take a break. I will resume at what is it? 9:48. Okay, if you think of any other questions, just leave it in the chat box, and we'll take a look at it. Okay, see you in a bit. Hello, let's keep going. So that's pretty much it for the blood stuff. Okay, now let's move on to lecture 3, which is the immune system, right, with the white cells and everything. Now what's missing in the blood review is the RH stuff, right? Like we spent a lot of time talking about it. So obviously it is important. So take the time to go through that concept, okay, if you're not familiar with it. So this is a table that has all the cells in it. I think it includes all the cells that we've learned. So let's just go through what kind of classification we can give them, as well as you know, what kind of function they do. All right. Just let me choose a better color here for the blue. Okay, there we go. So classification for macrophage, they are a phagocyte, meaning they are capable of doing phagocytosis, but more than that, after they eat up whatever they eat up, like a bacteria or something, they can do antigen presentation. So that's why they're an APC. Okay, antigen presentation cell, which is an important link between innate and adaptive immunity. Okay. And they can also be classified as a type of agranulocytes.
Is APC doesn't come under the function?
APC is a classification, right? Like, you know, is this cell capable of presenting? If it is, then it's an APC. So there are other APCs in the body, but this is the only one we've learned. Okay. So recognize PAMs on pathogens, and then they are able to perform phagocytosis. If you forgot what PAMs are, these are groups of chemical signals that are found on pathogens, right? And if a macrophage sees them,
Pathogen-associated molecular pattern.
that's right. And then they will eat them. They will eat anything that has PAMs on it. As an APC, they do antigen presentation, okay, to activate naive T cell. Okay, remember, they have to choose the correct naive T cells, which then allows them to do clonal expansion, right? Okay, next, neutrophils. Okay, I'm just switching colors to keep things like alternating, right? So like in case I run out of space or something. So this is agranulocytes. They are also phagocytes. Okay, but they're not APC. They just eat and they don't show the stuff to any naive T cells or anything. Okay, so main function, just phagocytosis. If they are the most abundant ABC, WBC, they are the most abundant. Okay, we'll say main function slash notes, I guess. I don't know what to call it. Okay, but just gotta put more stuff here. So most abundant WBC. 70% of the white cells are neutrophils. Okay, mast cells and basophils, they are granulocytes. And they play a role in what process?
Inflammation.
Yeah, trigger inflammation. Release histamine. Yeah. Okay. So mast cells. Mast cells is generally like localized inflammation. And then basophil is more like systemic, right? Bodily.
So the mast cell secrete the thromboplastin and this will secrete
No,
histamine.
they both secrete histamine. Yeah, they both secrete histamine.
Oh.
Yep. All right. NK cells. NK cells are natural killer cells, right? Not to be confused with killer T cells. Right, so these are part of the innate immunity. Okay, innate immunity. And they kill infected cells. Right, and they kill tumor cells. All right. Helper T cells, they are part of cell-mediated immunity. Okay, cell-mediated immunity. And you know, that's part of adaptive immunity. And they activate naive B cell. Help coordinate immune response. All right. Plasma cells. This is part of antibody mediated immunity. And that is, of course, also adaptive immunity. And their job is to mass produce antibodies. Mass produce antibodies. And what do antibodies do? They neutralize toxins and viruses. They form immune complexes. Okay, for easier phagocytosis. And they can also help activate complement proteins. Okay, remember protein antibodies are like Y shape, right? Y shape with a constant region and variable region. All right. That's all. Okay. Now there are other cells that is not in the list. I don't remember why I didn't put them on. I think I meant to put them on, and then I keep on forgetting to update the list every semester. There's suppressor T cells, memory T or B cells, right? There's also a killer T cell. Okay. So make sure you go through these ones as well. Are there other cells that we talked about that's not on this list? Did I miss anything? I think that's a pretty exhaustive list there. You know, obviously go through it yourself, add more if you need to. All right. How would you contrast specific and non-specific defense? For the interest of time, I'm just going to go through it with you. Specific immunity works only after non-specific have failed. Not necessarily, right? They could be parallel to each other. Non-specific defense and specific immunity requires prior exposure to be effective. Okay, to be effective. No, that's not true. Non-specific defense does not require prior exposure, right? Okay. And you know, specific immunity doesn't really need prior exposure either. Like you know, you could get it for the first time, but it just takes longer to recover. And then in the future, when you have the same infection again, then it works better. It works. It's more effective, right? B cells work in both non-specific and specific. Nope, only specific, right? B cell is part of adaptive immunity, part of specific immunity. Specific and adaptive means the same thing, huh? T cells work in both non-specific and also that's not true. Okay. Specific immunity changes throughout a person's lifetime, but non-specific defense does not. That's the answer. Okay, remember, non-specific are things like, phagocytosis, barriers to entries, right? Fever, inflammation, the fine things, right? And complement protein. These don't change. They are the same when you're born as it is when you are older. What changed throughout the course of your life is the specific immunity. As you get infected by different things, as you get vaccinated throughout your life, you gain these memory cells, you gain protections, change, it evolves, it adapts. That's why it's called adaptive immunity. Okay. Let's do number 10. I want you to do number 10. This is an important one. Okay, go for it. All right. We are looking for acquired immunity. Okay, so a RhoGAM injection is an example of artificial active immunity. Actually, none of these is correct. Okay, RhoGAM is not injected to treat anything, right? Like it's not injected to. It's not meant to create immunity. So this is like in the wrong context. Okay, RhoGAM is to prevent sensitization. And that is it. So both A and B are not correct. Okay. Nursing an infant to provide antibodies in breast milk is an example of natural active immunity. The problem here is the word active. Okay, the baby is not making their own antibody. The mother is passing it on to them. So that's why this is an example of passive. Okay, so this is wrong. Catching COVID-19 and recovering from it should give you natural active immunity. That is correct. Okay, natural, because you call it just through regular means, you didn't inject anything, and you have to make the antibodies. So that's why it's natural active immunity. Okay, so it will give you protection for the same variant, but if you get a different variant, you gotta do this all over again. Receiving an mRNA vaccine is an example of artificial active immunity. Okay, so D is the correct choice. All right, let's go through some of these things. Uses porphyrin to put holes in infected cells. That would be the killer T cell, okay, cytotoxic T cell. I don't know if I talk about porphyrin in the notes.
No problem. That was confusing me, yeah.
Yeah. So this is just like protein that puts holes, okay? But the point is they kill infected cells, okay, with these chemicals. So that is the cytotoxic.
That could be complement protein as well, no?
Name the cell though, right? Complement are not cells, right?
Protein is a cell as well, no?
No, protein is not a cell. Protein is not a cell. Cell is like, like alive on its own, right? Proteins are not, right?
Oh.
Yeah. Releases histamine to initiate local inflammatory response. That is mast cell. Mast cell. Next, a type of APC, antigen presenting cell that activates naive T cell. Well, if you have been with me today, and you know that is macrophage. Kills cancerous cells, NK cells, natural killer.
If we put monocyte, that will be wrong for macrophage?
For this one?
Yeah.
Yeah, no, it would be incorrect because neutrophils cannot present, right? Neutrophils can just eat.
Yeah.
They are not able to activate naive T cells.
Not the neutrophil, I'm saying monocyte.
Oh, monocyte, sorry. No, monocyte is like it's not activated yet themselves, right?
But those are the cells gets converted into the macrophage after differentiation.
Yeah, but it's like earlier in the steps, right? It's ultimately the macrophage that presents it to the naive T cells, right? Because if we all trace it back long enough, then they all go back to a stem cell, right? Like, you know what I mean? Yeah. So that's what we had to be.
But that will be multiple choice question, right?
But I wouldn't do that. I wouldn't put like monocyte and then macrophage in the same
Okay.
choice, right? Yeah.
If it is multiple choice, it's easy to pick because then it's there.
Yeah. All right. Form blood clots. That is platelets. Platelets. Okay. Most abundant WBC. That would be neutrophil.
Why is not fibrinogen instead of platelets?
Why is it not fibrinogen? Is that what you say?
Yes, Professor.
Yeah, fibrinogen is a protein, right? Right. This is name the cell.
Okay.
Mhm.
Right.
Name the cell.
Thank you.
Yeah. Okay. So like we've ran several iteration of this course online already, like ever since the beginning of the pandemic, right? Like so like I think this is our fourth year maybe, doing this online. And so the questions are pretty good now, I think. Okay, but for whatever reason, if you encounter a question that you are convinced something is wrong with it, or like it just doesn't make sense, you can let me know. Take a picture and send it to me after the test, right? And then we can I can take a closer look at it, okay, if that's the case. Because Mohammed, we were saying, like, what if the question has like monocytes and macrophage in it, right? Like, then which one do I choose, right? I would still choose macrophage because that's the most appropriate answer. But I'm saying, like, if you encounter a question, like you or anyone encounter a question that you think something is wrong with it, just let me know. It's possible. It's possible. Like I said, there are a lot of questions, and we have multiple people creating those questions. So yeah. And that's why some of you were telling me, right? Like some of the wordings is a little bit strange, okay? Because different people have different ways of asking questions, and that's why they don't all sound the same when you do them. Okay, now give examples of each of the following. Formed elements, well, we know it is RBC, aka erythrocytes, WBC, which is leukocyte, and platelets, which is thrombocytes. Okay, next we have granulocytes, which include neutrophils, basophil, and eosinophil. Okay, feel free to pop in a mast cell as well. T cells and B cells for lymphocytes. Coagulation proteins, that these are proteins that helps you stop the bleeding. Prothrombin and fibrinogen, right? Next, we have autoimmune disease. I mean, there are several. The one we talk about in class is Graves' disease. And you can, name any other ones, like multiple sclerosis.
Did we read the other one in our class, multiple sclerosis?
I think I talked about it briefly. It's like, I said this is when your T cell attacks your,
Oh, yes.
yeah, your neurons, right? Okay, physical barrier, skin, right? Mucous membrane, normal flora. Normal flora are the microorganisms that lives on us and in us, right? The harmless one, the ones that actually are beneficial to our body. Immunodeficiencies, it can be acquired like AIDS, or it could be something that people are born with, like SCID. SCID is the bubble boy, remember? Okay, you don't have to know a lot about them. Like it would be in a question would be like, which of the following is an example of primary immunodeficiency? Then you have to know is SCID. Right? Primary means you're born with it. Secondary means you're infected with it. You catch it later on in life. Lymphatic organs, spleen, appendix, thymus, tonsils. Example of artificial passive immunity. We talk about serum. Remember, like I got bitten by a dog and I need to get the serum for rabies. Natural passive immunity, breastfeeding. Okay, there are 4 characteristics of inflammation: redness, swelling, pain, and warmth.
Sir, I have a question, please.
Yes. Yeah, go ahead,
For
please.
8, can that not be a primary autoimmune deficiency if
Yes.
some
Okay.
babies
So
are born with it?
right. Yeah. So that's I understand what you're saying. Some babies, quote, are born with it, but not because of mutations in the gene, right? But because they caught it as they are coming out of the mother, who is who has the virus, right? That's what you're referring to, right?
Yes.
So in that sense, the baby caught it while doing delivery. It's when we say primary, it's something that is ingrained within their DNA, right? So in the case of SCID, it's because of a defect in the DNA. Right? That's what we mean. Right? Even though
So
we,
only when the baby's coming out, they catch it and not while they're in the.
yeah, so it's not like something that is within their DNA that caused them to inherently have the condition. You know what I mean?
Mhm.
Yeah, so that's what primary is, right? Like even
But
though the baby is catching it, like we say it's born with it, but technically it's like catching it from the mother, right?
But in today's world and society, since there's so much medication,
Mhm.
that the baby doesn't have to be born with it if they get the right medication at the right time.
You
Or would
talking
they
about
say?
you talking about HIV?
Yes.
Yes, it's possible to have intervention, I think, to minimize the risks. And even for people who like we talk about adults, right? Even for adults who suspect they have came into contact with HIV, there is a cocktail of drugs and prophylactic treatment that we can give to suppress the virus so that it doesn't actually, manifest into the disease. Okay,
Thank you.
yeah, good question. Um, Yes, because it's funny you ask, because I keep on telling you guys about these books, right? There is another book that I read that has talked a lot about this. And if you're interested, you can look it up, probably from your local library. I just got these things from the library, right? It's called Strange Bedfellow, and it talks about the medical aspect of sexually transmitted infections, right? Which includes a whole chapter on this HIV stuff. So, if you guys are interested, look it up. Okay, moving on. Why do we need to receive a flu shot each year in order to have protection against the flu virus? This was something that we talked about in the application questions of lecture 3, I believe. When I told you about the H1N1 and all those stuff. So the reason why we need one every year is because the dominant strain of flu virus that is circulating every winter differs, okay? Changes, so it's a prediction, and then we make a new vaccine. Okay, all the other ones are not true. People who don't believe in vaccines will think this one is true, but it's not. 14. We did this exact same question in the polls during class. The answer is B. You can go review that on your own. All right. So that is pretty much the third lecture, and we just did the respiratory system last week, so it should still be pretty fresh in your mind, I hope. But you know what? Let's go through it one more time. Why not? This is the graph that shows you the different breathing volume changes. So let's just go through it one more time. This thing here that just goes up and down is the tidal volume. Tidal volume. Okay, it just goes up and down, and it's just the inhalation exhalation change, right, when we're doing our regular breathing. If we inhale as much as we can beyond our normal respiratory volume, that is the inspiratory reserve volume. So the question here, A, asks, what is the expiratory reserve volume of this person? That's right here, ERV. Okay, so we can try to calculate it. Over here, that looks like 2,500. I promise if I give you a graph on the test, it will be a much nicer graph to get off. Like you won't have to estimate, but let's say this is 2,500. This here is 1200, I would say, roughly, right? So if you subtract it, it would be 1300 milliliters. Okay. So expect some calculation questions for this stuff. We did another one in the class, right? With the poll asking you to calculate the IRV or something. Right? Review that. What is the label for B? B goes from here all the way to here. Anybody remember what that's called? Yeah, that is the vital capacity. Very good. Okay, vital capacity. Describe the breathing pattern for the person. So just regular breathing. Okay, and then breathe out as much as you can. Back to regular breathing and inhale as much as you can. And then breathe out again. And then regular breathing. And then would, inhale as much as you can again. And then exhale all the way until you cannot exhale anymore. And then back to regular breathing. Okay. So hopefully that was straightforward. Describe the volume label as A. What is this thing? Anybody remember?
Residual volume.
Excellent. That is the residual volume. Another name for it is the dead air, right? That I keep on telling you about. This stuff doesn't come out. It's full of carbon dioxide. What condition causes the residual volume to increase? Do you guys remember that?
Smoking?
COPD, yeah, okay, that's part of it. But within COPD, there's another thing, right? That's okay. In emphysema, RV increases, right? Emphysema is the loss of elasticity of lung caused by loss of alveoli. In smoking, the alveoli fuses together and they form these gigantic air pockets that are no good for gas exchange. Okay. Draw a flow chart to illustrate how inhalation and exhalation occurs. So we actually wrote this down. I don't think I need to do it again for you. I'm just gonna refer you to the class note that we took. It's this one, right? So make sure you go through this again to know what kind of changes happens during gas exchange. Okay, the movement of the rib cage and the diaphragm and the change in the volume and all that stuff, right? Let me see if I can move this in. There we go. Okay. Right, so there are 4 factors that affects airflow, right? You should familiarize yourself with that as well. There's the Boyle's law. There is the pressure gradient, and the 2 pressures we're interested in is intrapulmonary pressure, as in the pressure inside your lungs, and atmospheric pressure. Okay, so when there is a difference between the two, the air will either enter the lungs or leave the lungs. Okay. All right, just a couple more questions here. During swallowing, what happens?
Larynx move upward to hit the epiglottis.
Yep, the larynx is gonna move up. Remember in class I told you to put your hand in the throat and swallow. You feel it pushes up, right? That's the larynx pushing against the epiglottis. Surfactant in the alveoli, that's the stuff that is produced by septal cells, and it basically breaks surface tension and increases compliance. Okay, it makes the lung more expandable. Okay, and it allows inhalation. Premature babies do not yet have their own surfactant, and it makes it difficult for them to inhale. The urge to breathe is primarily driven by, hopefully you know this by now, I've been repeating it quite often. It's an
Increase in
increase.
CO2.
That's right, increase in CO2. Okay, that is pretty much it. Do you guys have anything else you want to ask me about the test? Any other concept that you are,
Yes,
you know?
Professor.
Yeah, go ahead.
Professor, if you can describe the differentiation where the larynx starts and pharynx end in the skull picture
In the model?
in the model.
Yeah.
Not in the model, you know, there is a picture on the third or fourth slide which has all the parts.
Oh yeah. Okay, hold on. I gotta find that thing. Give me a second here. One second. There's also like something else I think I have here. Is it here? No, not this thing. Okay. Give me a second here. This one here? Or this one?
The previous one, Professor.
This one?
Yes.
Yeah, so like the lyrics is like, okay. Basically the lyrics is like where the Adam's apple is. Okay, like where you can touch your neck, right? That what you're touching is essentially the lyrics. Okay, the pharynx is directly on top of it. It's a really big area that is essentially the back of the throat. Okay, so you can see part of it in the mirror when you say ah, but it's like also higher up. Okay, like you won't even be able to touch it up here, right? Like when you do the COVID swab, you're swapping up here, but then like, you know, really further back, that's where the throat begins, right? Okay, so pharynx is located at the back of the head, and all the way a little bit back of your mouth as well, and that forms your throat in general. And then the larynx is just restricted in more of like the neck area. It's hard to find like a clear boundary, but if I have to, it would be like right below the glottis. That's where the larynx is gonna be. Okay,
Okay, thank you.
remember the glottis is where it leads to, you know, the larynx, right? The opening. Okay, so that is it. You know, good luck with the test. Next week we will start the new unit 2. The notes are already up. If you have any questions before the test, and even during the test, okay, like if during the test you have, you know, run into some trouble or whatever, right, just email me. Okay. All right. See you guys next week. Take care. Bye.
Thank you, Professor.
Yeah. No worries.